All right, I think we're going to get this webinar going. So today, Forum Bio is excited to chat with Andrew Carroll from Google about their advanced omics AI models, and also to talk a little bit about how we've integrated many of the tools Google Health has developed for variant identification in our Forum Bio platform. So I'd like to introduce Andrew Carroll, who will get us going to talk about all the cool things they've been developing over at Google.
Thank you, Brandy. All right, I'm going to try to keep a fairly punchy and hopefully interesting talk. The focus of the talk is going to be a background for what our team develops, and then 3 different stories about the sorts of things that these models that we're training learn from the data. So after that, then Chris Haas will give an overview of the Forum platform, and Brandy will have a Q&A session. Okay, so as I said, the focus of this section is going to be what can we learn from the way deep learning models train from genomic data. So at a high level, our team develops open source methods across pretty much every part of the sequencing lifecycle, from generation off of the instrument all the way through the combination with other modalities. You can see a list of the various open source technologies. I'm not going to go into all of these because I'm going to try to focus on a few stories here. The general theme for what our team does is it sits at the intersection of kind of large production data, the use of AI to train on that, and we have biological domain expertise to apply to the problem. So one of the key principles is that there's a variety of properties about sequencing and genomics data that we only partially or sometimes entirely don't understand. So there are unknown unknowns, the aspect of analyzing genomic data. So the approach that we use is deep learning, which can identify from a set of input data the kind of proper prediction that you would expect at a given position. So the key thing here is that you're learning directly from data. So the first of the stories that I'm going to tell is relies on DeepVariant. So DeepVariant is sitting at the middle of the process of analyzing data. Actually, yeah, a little out of order. So DeepVariant is a mechanism which looks at reads in a similar manner to the way people would expect to pile up in IGV, and it uses a convolutional neural net to identify the probabilities of reference, heterozygous, and homozygous variant positions. DeepVariant has won a variety of awards for highest accuracy across several different sequencing technology classes. And one important component is that DeepVariant's understanding of when it's likely to be correct or likely to be wrong is quite good. So this is a calibration plot with the variant quality estimated from the variant caller for DeepVariant and GATK. And if you're on that y equals x line, it means that you have a very good estimation when you're right, and you're confident that you're right, you're very likely to be right. And if you're non-confident, then you accurately assess the probability that you're wrong. And this goes back to the unknown unknowns aspect of the problem. The fact that there's some things that's very difficult for us to programmatically model. And so methods that train from data directly have a much better chance of identifying and understanding all the factors that are involved in them being correct or not. So DeepVariant is something that we have put out a variety of models for over the years. We've extended it for multiple sequencing technologies. We've extended it for multiple problems like exome sequencing, for example, or trio calling, or the use of pangenomes to map to. So it's something we stand behind as open source software. And we're very happy that Forum has taken DeepVariant and actually all of our methods, including DeepSomatic, DeepConsensus, DeepPolisher, and others, and put those onto the platform to make them easy to use. One of the aspects of being able to learn directly from sequencing information through training is that you can very quickly adapt to new technologies. So in the last couple of years, there have been a variety of new sequencing technologies that have come out. And because DeepVariant can learn directly from that, it's very easy for us to put out new models. So these are just some examples, and I'm showing the Ultima Genomics white paper that they have of using DeepVariant to have highest accuracy on the data sets that are present. Another example Another example is the work that we did for Oxford nanopore sequencing with Stanford University, where they were able to set the record for the fastest time to diagnosis. This was the first case where we're able to show that a nanopore-based pipeline could analyze a genome actually as or more accurately than Illumina data. And since then, there have been a variety of demonstrations that we can now push the accuracy of nanopore fairly high. And in this case, I did a pilot for 12 individuals, of which we diagnosed about half of them, and all of them in record time, some of them less than 8 hours. Okay, so now I'm going to switch to a section of talk where we're going to go through some examples of the components that the deep learning model has learned that were maybe a little bit surprising or interesting to us. So one of which is this concept of segmental duplications and copy number variants. So we received some bug reports early on that said, why isn't DeepVariant calling some of these things that look like variants in my data? So for example, if you have these reads that are mapped to GRCH37 and you look at this position, I think most humans would say this looks like a heterozygous variant. And maybe it looks like a fairly easy call at first inspection. And DeepVariant actually says this is not a variant. And so we got some reports of this, and we started taking a look at exactly why or what is going on. We're trying to find a bug, and we can't find any bug. And things become a little bit clearer when we take the same reads and we map them to GRCH38. So this position looks fairly different in GRCH38. So now you see you have this one heterozygous variant remaining, but all these things that looked like variants before don't show up in the reads anymore. So what's going on here? So it turns out that this position is some sort of a collapsed duplication in GRCH38, where there's another copy of this gene that actually belongs somewhere else in the genome. That because GRCH38 is a more recent genome build, it's represented in 38, and those reads have a home to map to, but it's not in 37. So this is effectively, these things that appear to be variants are actually effectively a copy number variant in the gene relative to the reference. And in the process of training, DeepVariant was able to learn this concept and likely is able to use the phasing between these variant blocks to figure out that there's actually 3 copies here and which should be present. But it's quite interesting because DeepVariant was never specifically trained for copy number data. So it learned that property from the data itself. The second story is this new and emerging technology of using pangenomes, which has been very exciting to us. So the pangenome is a graph representation of the reference. So you can have multiple different individuals' complete sequence instead of the prior linear reference, which is a set of pieces of individuals that are sort of merged together and at some places don't have a sequence which appears in any single individual. So the Human Pangenome Reference Consortium, which we're actually working with in a variety of forms, from the data generation all the way to like filtering and improving the quality of the assembly and then using the pangenome, is something which aims to build new reference resources. So one of the things that we can do now is we can train DeepVariant on the pangenome, and we can provide the information from the pangenome to DeepVariant, and we can see that this will substantially improve the general accuracy of DeepVariant, and especially in other processes like structural variant calling, using a pangenome is quite a bit stronger. And one advantage is as well that we have a method which will then put the calls back onto the 38 reference coordinate system. So this will just generally improve the accuracy even on the files that you're familiar with. So one specific example of a gene that's improved, there's this KCNE1 gene, which is associated with arrhythmia. And you can see in these plots what you get with DeepVariant, with the pangenome and with the linear reference. So with the linear reference, there's a bunch of positions which can't be mapped. So you effectively miss these positions. But with the pangenome, we can now map these and put them into the correct position and now call them correctly. So this is just one example of the tricky regions of the genome that the pangenome is very helpful for. The second story I have is, the third story I have is with insights from the RNA model. So basically, we're able to train on a bunch of RNAseq data and get a substantially more accurate. This is, I think, about a 50% error reduction relative to the prior state of the art. And this is one of the models that's on FormBio, one of the models you can access generally. It's a RNA production model. But the interesting things I want to talk about are a few things which drive this improvement. So one of the things you can do is you can look into regions and try to identify why is this model better than, say, our standard exome model or some other methods. One thing that you identify pretty quickly is, so this is the DeepVariant RNA model here, is that there's a much lower error rate at exon boundaries. So the DeepVariant model has learned in the process of training some concepts from splicing and been able to more accurately use them to call variants into positions. The second is that the model, despite actually not being trained at all, has learned the concept of RNA editing. So RNA editing manifests as something that looks like a variant, specifically looks like these A to G or T to C variants, but it's actually not. It's a post-transcriptional change to the sequence. So if you use the exome model, which is only trained on exome data, you'll have this large number of false positives that go away when you train with the RNAseq model. And in order to get this reduction in errors, the model effectively has to learn to predict what is an edit site. So even though we didn't have any training data for RNA editing, you can see that in this database of known edit sites, we substantially reduce the error rate. And this actually gives some mechanism to predict an edit, because you can run the exome model and you can run the RNA-seq model and look at these positions for where they diverge in their prediction. And those are sites which are putative edit sites. So those are the stories. I went through them relatively quickly. Hopefully they're interesting to folks. This is the team and the breakdown of people. The DeepVariant and DeepConsensus team are these folks. And then these folks here do other work, which I didn't have a chance to talk about, combining genomic and non-genomic data. And with that, I will hand it off to Chris.
Thank you, Andrew. Just let me bring my screen up. Thank you for attending, everyone. My name is Chris Haas. I have two hats at FormBio. Well, probably more than that, actually, when I think about it. But the two main ones are I lead product management for our platform, and I also lead up partnerships, which is heavily focused on working with Google, Andrew Carroll and team, and Google Cloud as close partners of ours. So I want to quickly talk about our a few slides on our GCP Marketplace product, where our platform is hosted, and the collaboration with Andrew and his team on the Google Research Genomics team. So first, just about the Marketplace product. It is a self-service, self-provisioned product available in GCP Marketplace, very easy to find. I'll show you our landing page, and I'll jump into the Marketplace real briefly, but it is a less than 10-minute process from time of discovery, subscribing to one of our plans, and then provisioning your organization and project within FormBio. Very simple, straightforward. I do want to make a call out on this, that Google Cloud gives every new Google user to Google Cloud Platform $300 in free credits that you can use as part of your evaluation of the products on Google Cloud Marketplace. So Google, we call it Deep Omics. This is our collaboration with Andrew and the Google research team focused on genomics at Google. We work very closely with them to implement all of their DeepX algorithms that they have open source. So DeepVariant, like Andrew has discussed, DeepConsensus, Deep Semantic, Deep Trio, Deep Polisher, and we continue to work with them closely we'll onboard their next DeepX that they launch into our platform as quickly as we can, so that our users can actually take advantage of that in a point-and-click fashion. Another just tidbit about the collaboration is Andrew and his team have closely worked with us to benchmark our workflows to make sure that it meets their performance and quality requirements that they would expect for anyone using their open source code and how they benchmark their own algorithms. So what do we offer? High-level functionality. First of all, we really wanted to make a platform super easy to use. It supports the power users like the bioinformaticians, but also can support a bench or lab scientist who just wants to go in, not necessarily work on a command line, scripting things away. Just they want to kind of point and click through an easy to use interface to upload their data, run a workflow, get the results, and move on with the research that they have. So data management is a key area we focus on, trying to make it very easy to get data into the platform, manage that data, and then launch into workflows. We have a lot of workflows that we have developed and put onto the platform. You can see the various categories we have. Specifically calling out the Deep category, which has all of the Google algorithms that we've talked about already. I'll show some of those. And I also want to call out that we allow you to bring your own workflows onto the platform. We support all of the NF Core workflows out of the box. We support also, and I'll briefly show this, the ability to connect public and private GitHub repos. So you can bring that those open source, if it's public, or your own private workflows onto the platform and run those as well. Data visualization is another area we really focus on, trying to give you just out-of-the-box visualizations that you would expect and find useful so that you can really get directly to the insights that you want out of the workflow results and the data that you've put onto the platform. And obviously, you can take the data with you and bring it into whatever other visualization tools you want. And we continue to actually enhance this with other visualizations we embed in the platform with support also for bringing your own. Collaboration, another key part. We know all of you want to work together as researchers, share data, share results, share insights. And so we want to make it very easy for you to collaborate on the platform with all of your users. Let me switch over quickly to our landing page. So formbio.com/start, real easy place to just come in, quickly review high-level information, some of which I just showed you, and then go straight out into the marketplace. And more similar information, more details. Obviously, you have the plans that are available to you. We have an individual plan, which is designed for someone who doesn't want to, it's subscription free, so you can just sign up and then you just pay for your own usage that you do on the platform. So any data you upload, any workflows you run. If you are involved with a research team, small or large, and you want to have other functionality and support many, the whole team, you have the team plan and the pro plan. We also can work with you on what's called private offers in the marketplace if you have other more specific needs that don't necessarily come out in the features here, or you want to kind of piecemeal things together as far as the functionality that you would prefer. All right, so let me jump into the platform itself. So first, jumping right into the homepage, just give you some real high-level things to do straight away. Normally, you want to add data to the project. That'll be the first thing I jump into once I review some other things first. We show you an activity log of all the workflows that have been run within this particular project. So we do have the concept of an organization, as well as projects underneath that organization. Within the project, you can have your team members, and then you will see the activity of what the entire team has done in this project. We obviously embed some news and videos about what's happening with FormBio, tutorials, updates, things like that. Adding users is very easy. Just go to the Members tab, select the Add Member, and then enter their email, choose what type of role they have, and they will get an invite to come onto the platform into that project and collaborate with you. I do want to call out before I jump any further is I'm showing you the interface here due to the webinar. We do have a command line client, an API, so that you can have more of that programmatic or command line interface to the platform and script to your heart's content as far as loading data onto the platform, retrieving data from the platform, running workflows, getting the results of those workflows, etc. So for adding data, there's lots of ways. So command line I mentioned, so you can just add data directly through the command line from your local computer, as well as other sources, depending on where you're running the command line client. We do have a data transfer tool that I'll show that supports lots of out-of-the-box data repositories, including all the big blob storage across the 3 big cloud providers, Google, AWS, and Microsoft, as well as others like Oracle, and then personal drives like Google Drive, OneDrive, etc. I'm going to take a different approach for adding data once I show you the data tab. And so it's just a different alternative to pull data from public data sources. So first thing is data. Obviously, I just want to add data. I can drag and drop command line client again. It will all show up here and be organized based on the folder structure you choose. And I can move data around. I can see what data I have and choose what I want to do with that next. Generally, I'm going to want to run a workflow against that. So in the workflow tab, you see all of our categories. You can see we have lots of out-of-the-box workflows that you are able to use. These are our opinionated based on our Brandy's team, the scientific experts we have, knowing what researchers very commonly do, developing workflows to support that. Specifically around Google DeepOmics, just to give you a view of like what we have on the platform, we have the Google algorithms embedded into workflows in a couple different ways. Standalone, just, hey, I just want to run DeepVariant. Great, you have a workflow for that. I just run DeepVariant, get my results, and move on. And then we've taken, like I've said, we have these opinionated approach of how we want to implement specific types of workflows aligned to scientific use cases, such as expression analysis with RNAseq, where we've embedded DeepVariant for RNAseq as an option within that workflow. Before I jump into that, I want to pull in some external data. So I'm just going to pull in some public data files from SRA. So quickly, and I'll show this again, so I'm not going to go into too much detail, but we always start with an overview page of what the workflow does. You have version control, so when we launch new versions, you see updated versions. You can always go back to a prior version if you want to stick with the one that you had. Run this workflow, short read. I'm going to select an existing sample file. I'm going to do a cancer example here. I could have just created my own sample table if I wanted to. I could edit this table and save it back out if I need to make changes before submitting it. This is a very simple workflow, so it's just, give it a name, and I'm off and running. I already have this one pre-run, so I can show that. So my activity page, similar to what we showed on the home page, all of the prior runs that have occurred. So here's the prior run I ran for that cancer RNA-seq download from SRA. So you can see all the files that were pulled down across the various samples for control and test. And every workflow looks the same as far as the result page. I have a status, so I can see every step that occurred, whether it completed or not. These all were successful. If it was an error, it would show me an error here, and then I can expand and debug to figure out what's going on. And I can link out to each one of these files as needed to do that debug. I have my All Files tab where I can actually see all the results. So there are my FASTQ files that I pulled down. I can see the inputs that were run for that execution of the workflow. So I can, if I'm collaborating with someone, they can see what my settings were and understand like what I was doing. And then obviously we have our Logs tab, which shows everything in a more detailed view as far as the logs for that workflow. All right, so I have my data now. So I wanted to go back in and do my expression analysis. All right, so more detailed view of the landing page. So this is obviously a much more robust scientific process workflow. So we really want to detail to you what it is doing across various configurations that the workflow has available to you. So you can see all the various options here with what the execution tree structure looks like. We're giving you example outputs in comparison to other implementations of this type of workflow. So you can just quickly see how we would compare with ours versus someone else's. Citations, very important, obviously, for the scientific process. We want to make sure you understand where this workflow came from. Like all the various computational steps, we will have a citation for where that research was done, that code was produced from, and you can refer to that. Inputs. Just before you launch, what's it take to actually run this workflow? What inputs do you need, et cetera? Outputs, showing you the results, and then we really like to call out the citation. So we have that separated out in its own citations tab for you to refer to. All right, so let's run this workflow. Okay, so I'm gonna do a full analysis. I am doing a human cancer sample. Select a directory. Professor Brandy, help me. I forgot where I was supposed to go. By the way, everyone, I am a technologist. I have an undergrad in biology, but I am not a biological scientist. So I always rely on my friends Andrew and Brandy.
So we want to go to pipeline outputs,
Pipeline outputs. Okay.
and then
Oh,
go
there
down.
it is. Get NCBI, right?
Yeah, that's right. And then the
And then,
last
so these are 2 runs that we did of this workflow. So I'm going to select my latest one.
output. And then
Output FastQ. There we go. Thank you. And I'm going to select an existing sample sample sheet.
It's going to be in the same folder as the data.
All right. Thank you for that help. There we go. All right, so let that load. There we go. Again, I can make changes to this if I wanted to, and I can save it back out. Move on to the next step. Choose the latest as far as the reference genome. We do allow, so we have a lot of public genomes on the platform. We also support private genomes. So we don't have any private genomes in this demo project, but if we did, you would have a private genome section that you could select here based on the private genomes you've uploaded into the platform. Let's move on.
And just to say, what a private genome is like, let's say you sequence the unicorn genome. You wouldn't want to share that with the world yet because it's too precious and glorious. And so you might add privately your unicorn genome and then do your RNA-seq analysis against your own private unicorn genome. But this is a human cancer, so obviously the genome here is going to be human.
Thank you, Brandy. Okay, so my advanced configuration, you can see lots of options that we give you. This is a fairly robust workflow. I'm going to run this with DeepVariant, but you can see other options I have available to me to tweak the way I want this workflow to execute. Once I'm done, again, just give it a name. I can see what I've my settings. I can go back and make changes if I needed to, and then I would just execute that workflow. So let's go see what the results look like. So again, I had this one run already. All right, so here's my results. I see all my files. We do give you a QC output for the workflow run. Just very easy, quick review of common visualizations that you would want to see for the execution of that workflow. Scroll down and show you some of those real quick. Again, I kind of talked about the other tabs. So my workflow execution, did I have any errors? I can see all my results. So some other things that we have embedded in the platform. One, IGV. So that is something that you have embedded in the platform that is available to you for the appropriate file types. You can open that. No need to have IGV installed somewhere else. That's available for you. We also have hosted RShiny applications that we have built. And that will show up as an open analysis tab here, which I will show here. I already have that open. So just a more detailed analytical application for you to look through the results and really gain those insights. So quick call outs here, things like, I can see a really nice separation between my control and sample sets. I can see other visualizations that are provided to me through the application. And again, I have my inputs and my logs. Okay, so that would 2 workflow examples. Obviously, there's a lot more we could go through. I do want to call it a couple of things. One, the bring your own capability. So under workflows, you have this manage option that allows me to connect to GitHub repo. Then once I have that connected, I'm able to bring in any, this is a private repo for the demo, but I can bring in any of the repo repositories that I have in that private repo. I can obviously connect a public repo to bring in anything from there. This is where we would start bringing in, we have NF Core more natively integrated into the platform where you don't have to do it through this mechanism, but if you had another public GitHub repo, for example, you wanted to pull in DeepVariant directly into this for some reason, not use our workflows, you could do that through this mechanism. Some other things to call on the platform. So back to like things that we try to give you as options onto the platform to allow you to just really advance the research you're doing around visualizations, around data transfer. we have this idea we call Form Bench, which is hosted applications on our platform. Quick callouts, data transfer. So let me open that one first. That would be a way to launch it. I actually have one running already. So this is the mechanism I talked about, where you have lots of options to pull in data from public resources. So you can see I have a couple pre-configured data sources, just to kind of show you an option. So here are a list of what we have available. I'm going to go to select AWS S3. I have all the configuration options that I need to pull in, to connect to the buckets I have in S3. I'm not going to go through all of those right now. Once I have that configured, then it's available to me to browse and then transfer data into FormBio. Other options we have a form shell if you want to be on the command line, hosted command line client. We have JupyterLab, RStudio embedded, and then some other more advanced visualizations like cell by gene and others. You do have the ability to import your own form benches. So here's a couple examples at the bottom, and I would do that through the manage option under form bench to import a Dockerized application that then we will host for you and manage the execution and running of that on FormBio. And just to show an example of JupyterLab, I just opened that running JupyterLab instance that I had, and it's the familiar JupyterLab environment that you expect. We do actually mount we mount all the data in that project that you're launching this from, so that you have all of it available for you to go in and develop your JupyterLab scripts to gain the insights that you need. This comes pre-configured with lots of packages already pre-installed. Same with RStudio. So we've gone through and kind of built a curated curated instance that incorporates most of the libraries that are commonly used. You can bring in your own as well to extend it even further. All right, I know we're kind of running fast. Hopefully there's some good questions that have come in while I've been presenting here. Brandy, anything that you think I've forgotten that I should call out before I switch back and open it up for Q&A?
I don't think so, Chris. I think you showed, yep, you showed everything. Maybe you want to just show quickly the documentation and how people would get support.
Yeah. And that actually reminds me, we do have this really simple, easy to use notes tool that is across the entire platform. It'll get, when you create a note, it gets tagged for where you were in the environment. So if you're in a specific workflow, it'll get tagged for that. And that's just real simple, easy way to collect your thoughts as you are working with the platform, and then have those thoughts available to other team members. We have docs. This is a external site, so you can launch out to it. Internet's running a little slow, so it's not showing up right now. There it goes. But you also have it available down here, which will actually launch out to our docs site, which has information about all of our workflows, the platform itself, overviews. You have a way to contact support, so you can get a hold of them if you have any questions. I also want to call out our FormBio Academy, which we will continue to push videos out to. That's something we put into the homepage, so you have the videos available, but this is publicly available that you can jump out to and see these videos created by our bioinformatics team. All right. With that, I'd like to open up for questions and answers, and you should be receiving an email. If it's not in your inbox already, it should show up soon from our marketing team with details about everything we presented here, and like next steps you could take if you wanted to go and evaluate us. The quickest way is to go to formbio.com/start. Here's just an overview of the published plans we have. If you do have more unique needs, you can always contact us. And we can customize a plan for you if one of these don't fit your needs. quickest way to get started is that academic individual tier. It's not just for academics, it's really for anybody. It's just a plan with one user, subscription free. You just pay for whatever usage you have or that you've done. So whatever data you upload, what workflows that you've launched, and you just pay through the marketplace. You would pay for that using whatever billing ID you create when you go and subscribe to us.
All right, folks, if you have some questions, put them in the question chat. Can team users share notes, Chris?
I believe notes are available across the entire. So in that project, all team members will see the notes. So if you create one, it'll be tagged for where you were, who it was, and then those notes will be visible to everybody. So you could collaborate on those.
So inquiring minds want to know, Andrew, when are we going to see a deep structure or a deep copy?
Right. So that's an interesting question. I think our current plan is to go deeper into the pangenome component of it, and I think in the long term we see variant calling kind of merging in with this concept of assembly polishing, right? That at some point you have a very comprehensive pangenome, and then you're like correcting errors relative to it. And when you do that, the copy number and structural variants sort of naturally emerge from the comparison with the pangenome. So I think that our plan is not necessarily in the short run to have a traditional copy number caller, but maybe in the longer term to go more on that pangenome side. But one thing that might change that, and an interesting thing is that our team often does 20% projects with other people at Google. Like we have folks who work in ads, for example, like work 20% time. And if there's a 20%er who's interested, this actually sounds like a really good kind of like 20% project where we structure a little bit and then they kind of take it. And there have been some of these things, some of our outputs have like emerged from those sorts of projects.
Yeah, that sounds really cool. I mean, I definitely feel like you're right with normal, let's say, inherited genetics. Having a good pangenome is key, but in cancer, for example, you don't know what sort of crazy things are going to happen, right, to your genome, with aneuploidies and such.
Yeah, that's fair as well. I mean, actually, for cancer, the interesting thing is like what you do within normal sequences, which you can like base all of your calculations relative to that.
Right. Yeah. What sort of sequencing technologies does DeepVariant take?
Right. So we try to support pretty much every technology. Like one of the things I mentioned is that because it's very easy to train a model. So when a sequencing company reaches out to us, often we can get them a starting model within a week of them first giving data to us. So we have models that are good to go for Illumina. We have whole genome and exome model. We have models for Element. There are models for singular genomics. There's models for Ultima, although I think Ultima hosts the models themselves. We have 2 examples for complete genomics, and we have PacBio as well as Oxford Nanopore. So I think that's pretty much everything. If there's something we're missing that you really want us to develop, let us know. And if you can get us genome in a bottle data, like I said, we can usually get a model out in about a week.
Great. Chris, there is a question about a quality assessment. Maybe we're going to go back to the platform and just show a couple of our quick tools, particularly the FastQC and Kraken. So these are tools where users can quickly assess sequence quality and contamination, and possibly find, which we found, with some of our customers, cases of bad demultiplexing or sample swaps, where you think it's supposed to be mouse and actually all your sequence is human or vice versa. So you can find that on these. So we have these quick tools, which are small little tools folks can run. And if you click on Power Tools, which is where I think we put our QC, we find our FastQC and our Kraken on these tools. Any other questions from the audience? Just give it another minute.
Randy, since I didn't really call it the quick tools, just want I don't know if you just described them enough. Just they're really designed for very quick, more or less single step sort of quality or quick step tools versus a more full-blown workflow, right?
Yeah, that's right. Like they're single. So the difference between a power tool and a quick tool, a power tool is a requires quite a bit of resources, but it's run on a single machine. So it's like the idea of a one-step workflow, which is why you see, Deep Somatic and DeepVariant there. And then a quick tool is something that should run in less than 10 minutes on a single node machine and not require much more than a gig of memory. So those are, sort of, simpler tasks like aligning two sequences or two structures or, some sort of sequence calculation, data extraction or download, things like that. So that's our quick tools and our power tools.
And since I didn't really do this before, I just want to call more specifically the end of core workflows. So we've more natively built that in versus connecting the GitHub repo. As you can see here, how we support all of the available NF core workflows just natively in our workflow interface.
Great. So it doesn't look like there's any other questions from our audience. So I think we'll be wrapping up. Thank you everyone for joining our webinar on the FormBio platform and the implementation of Google's Omics tools on our platform. And, look out in your email box for invitation to try out the platform with, all the information that you might need to get going. So thank you so much, everyone. Thank you, Andrew. Thank you, Chris. And have a great day, everyone.
Thank you, everyone.